Welcome to the Learning Module for Basic Genetics and Genetic Diseases. This is Dr. Tafke for the Cellular and Molecular Medicines course. After studying this learning module, students should be able to compare the types of genes normally giving rise to recessive and dominant acting disorders, explain dominant negative mutations and give an example, explain haploinsufficiency and its role in acute intermittent porphyria and familial hypercholesterolemia, describe the major modes of inheritance, determine the likely mode of inheritance from a pedigree analysis, describe the characteristics of trinucleotide repeat disorders, explain the phenomenon of anticipation, describe the molecular mechanism underlying Huntington's disease, Fragile X syndrome, myotonic dystrophy, and Friedreich's ataxia, explain reduced penetrance, variable expressivity, germline mosaicism, and pleiotropy. Explain genomic imprinting with regard to Prader-Willi syndrome and Angelman syndrome. A recessive allele is one in which only the homozygous recessive individual displays the phenotype, and heterozygotes are phenotypically indistinguishable from wild types. The underlying molecular basis for recessive acting mutations is that they typically occur in enzymes or proteins that function alone as monomers. In other words, the protein products of each allele function independent of each other to produce the phenotype in the cell. In the case of monomeric enzymes, the net enzymatic activity in the cell is simply the sum of enzymatic activity of the proteins produced from each allele. If a single wild type or good copy of a gene is sufficient to produce enough protein or enzyme for the cell to function normally, the phenotype is unaffected. This condition is known as haplosufficiency. Even though heterozygous individuals are phenotypically indistinguishable from homozygous wild types, they can often be diagnosed because of reduced enzyme activity. The same is true for genes that code for structural proteins that function independent of each other. If a heterozygous individual can get by with a reduced level of normal protein produced from the wild type allele, the mutation is acting recessively. Only in the homozygous condition where both alleles carry the mutation does the lack of enzymatic activity or protein function result in the disease phenotype. Dominant mutations, on the other hand, typically occur in genes coding for proteins that form multimeric structures in order to produce their effects. In the heterozygous situation, the production of wild-type protein from the one good allele is not enough to produce a normal phenotype. This is referred to as haploinsufficiency. In other cases, the production of the mutant protein results in a diminished capability of the cell to assemble enough functional protein complexes for the cell to achieve a normal phenotype. The presence of the mutant protein disrupts the function of the complex, or in some manner antagonizes the function of the wild-type protein. This situation is called a dominant negative mutation. The homozygous situation can result in an even greater loss of function. Heterozygotes for dominant diseases are rare to begin with and often too sick to produce children. For most cases of dominant acting disease alleles, a homozygous child of heterozygous parents is much more seriously affected than the parents. In these cases, the dominant allele is actually incompletely dominant. For example, persons who inherit a defect in the fibroblast growth factor 3 receptor have achondroplasia, which is a form of dwarfism. They usually have a fairly normal lifespan. Homozygous children are much more severely affected and usually do not survive infancy. Mutation in a number of genes can lead to polydactyly. Heterozygotes generally have extra fingers and toes. The homozygous children of heterozygous polydactyl parents often have severe developmental defects and do not live past early childhood. Familial hypercholesterolemia is caused by mutations in the low-density lipoprotein receptor. Heterozygous individuals have elevated serum cholesterol levels and develop early heart disease, usually by the 5th decade of life. The homozygous individuals have even higher serum cholesterol levels and usually have MIs and strokes in their early 30s. On the other hand, Huntington's disease, caused by mutations in the Huntington gene, acts completely dominant, as heterozygotes and homozygotes are affected equally. There are 3 broad categories of genetic disorders. Multifactorial disorders are polygenetic. They have many genes that influence the phenotype. They are also controlled or influenced greatly by the environment. They can show patterns of inheritance and run in families, but it's often difficult to determine exactly what the risk is of passing on these disorders to their children. A good example of multifactorial illness is essential hypertension. Chromosomal abnormalities, including chromosomal rearrangements and conditions of polyploidy or aneuploidy, for example, inheriting an extra chromosome 21 results in trisomy 21 or Down syndrome, and monogenetic or single gene disorders, which are usually classified by their mode of inheritance as either autosomal dominant, autosomal recessive, or sex-linked. The degree to which any gene or environmental factor can contribute to a multifactorial disease can vary greatly. Some can be 100% genetic, such as Down syndrome or achondroplasia. Others can be 100% environmental, such as being struck by lightning. Others have varying degrees of genetic and environmental influence. And some, like PKU, are 100% genetic, but the damage done results to exposure to an environmental agent, phenylalanine, in the diet. Can vary greatly. Single gene disorders are transmitted in a Mendelian fashion and are classified as either autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, or Y-linked. There are relatively few autosomal dominant human diseases, but some rare traits are inherited in this fashion, most notably achondroplasia, which is a form of short-limb dwarfism caused by a mutation in the FGF3 receptor gene. Autosomal dominant traits will typically be present in every generation of a family pedigree. An autosomal dominant inheritance pattern will show both sexes affected equally and that both sexes can transmit the trait. It will not skip generations. Importantly, all affected offspring will have an affected parent. When one parent is affected and the other parent is unaffected, approximately half of the offspring will be affected. And importantly, unaffected parents cannot transmit the trait. With autosomal recessive disorders, affected individuals are homozygous, having inherited one disease-causing allele from each parent. The parents did not have the disease, but were carriers of the disease-causing allele. Examples of autosomal recessive disorders are cystic fibrosis, sickle cell anemia, phenylketonuria, and Tay-Sachs disease. Autosomal recessive diseases are typically only seen in one generation or in skipped generations of a family pedigree. An autosomal recessive inheritance pattern will show that both sexes are equally affected and that the trait skips generations. Affected children are usually born to unaffected parents. And when both parents are heterozygous, approximately 25% of their children will be affected. Genes inherited with the sex chromosomes are called sex-linked. Genes carried on the X chromosome are known as X-linked. X-linked dominant diseases are really rare. They are often neonatal lethal in males and only present in heterozygous females. X-linked hypophosphatemia ranges from isolated hypophosphatemia to severe lower extremity bowing, causing a rickets-like phenotype. Incontinentia pigmenti is characterized by skin abnormalities that evolve throughout childhood and young adulthood. Many affected infants have a blistering rash at birth and in early infancy. This heals and then is followed by development of wart-like skin growths. In early childhood, the skin develops gray or brown patches, hyperpigmentation, that occurs in a swirled pattern. Inheritance patterns for X-linked dominant disorders will often show more females than males are affected, and it will not skip generations. Affected sons must have an affected mother. She is who gave them their only X chromosome. Affected daughters must have either an affected mother or an affected father. All daughters of an affected father will have the disorder. Affected mothers, if heterozygous, will pass the trait on to half their sons and half their daughters. With X-linked recessive diseases, all males carrying a recessive allele on the X chromosome will be affected, while only homozygous women carrying two recessive X-linked alleles will be affected. Occasionally, heterozygous females will express an X-linked recessive disorder due to random inactivation of the X chromosome carrying the normal allele during X inactivation, which occurs early in development. These individuals are referred to as manifesting heterozygotes. The disease expression in these individuals is usually milder than an affected male, as they typically have at least a small population of active X chromosomes carrying the wild-type allele. Some well-known X-linked recessive diseases and traits include hemophilia A and hemophilia B, Duchenne muscular dystrophy, and red-green color blindness. Inheritance patterns for X-linked recessive disorders will often show more males than females are affected. Affected sons will usually be born to unaffected heterozygous carrier mothers, so the trait will skip generations. Approximately half of the sons of a heterozygous mother will have the disorder. Since fathers only pass a Y chromosome onto their sons, X-linked recessive diseases are never passed from father to son. All daughters of affected fathers are carriers, and they may be manifesting heterozygotes due to skewed X inactivation. Mutations in the mitochondrial genome can give rise to mitochondrial disorders. Mitochondrial disorders have two unique features. While both sexes can be affected, only females can pass it on to subsequent generations. This is because only the egg contributes mitochondria to the embryo. Thus, all offspring of an affected female will have the disease. A second unique feature of mitochondrial diseases is heteroplasmy. Cells can contain hundreds of mitochondria, each with its own genome. If every mitochondrial genome in the cell carries the exact same mutation, it is homeoplasmic. However, if the cells contain a mixed population of normal and mutant mitochondrial genomes, there is heteroplasmy. Variations in the heteroplasmy means there is variation in the severity of expression of the mitochondrial diseases. A fairly new type of mutation has been described in which a particular 3 base pair expansion in a gene causes disease. These disorders then are collectively known as trinucleotide repeat disorders. During replication, DNA polymerase can stutter when it replicates several tandem copies of a short sequence. In trinucleotide repeat disorders, the number of repeats correlates directly with the severity and onset of the disorder, and the length of the expansion can increase with every subsequent generation, and the severity will increase as well. This is known as anticipation. They can be broadly grouped into poly-Q disorders, which have a CAG repeat, with CAG being the codon for glutamine, so these disorders produce a polyglutamine tract in a protein, and non-poly-Q disorders that have another trinucleotide repeat or a CAG repeat located in a non-coding region of the gene. Some well-known trinucleotide repeat disorders include Huntington's disease, which is caused by CAG repeats in the Huntington gene, Fragile X syndrome, which is caused by CGG repeats in the FMR1 gene, Friedreich's ataxia, which is caused by GAA repeats in the frataxin gene, and myotonic dystrophy, which is CTG repeats in the DMPK gene. Huntington's disease is a trinucleotide repeat disorder that causes a progressive breakdown or degeneration of nerve cells in the brain. It is caused by an accumulation of CAG repeats in the Huntington gene. Neuronal degeneration occurs, leading to progressive dementia, evolving involuntary movements, and psychiatric disturbances. In normal individuals, the number of CAG repeats in the Huntington gene is less than 35. In affected individuals, the number of repeats can range from 35 to well over 100. And the longer the expansion, the earlier the onset of the disease. Fragile X syndrome is caused by an expansion of CGG trinucleotide repeats in the promoter region of the FMR1, or the Fragile X mental retardation 1 gene, on the X chromosome. Most unaffected people have only a couple dozen CGG repeats in the FMR1 gene. Individuals with Fragile X syndrome have accumulated greater than 200 of these CGG repeats. This results in a hypermethylation of the gene promoter, silencing it. Shortage of the FMR protein then disrupts normal functions in nerve cells, causing learning problems and intellectual disability and other symptoms of Fragile X. Fragile X gets its name from the appearance of the mutated X chromosome, where the ends of the Q arm are constricted by the mutation, giving the chromosome a fragile appearance. Friedreich's ataxia is caused by an expansion of GAA repeats in the first intron of the frataxin gene. In normal people, the GAA repeats in the frataxin gene is less than 33. But in people afflicted with Friedreich's ataxia, the number of repeats can number in the thousands. This greatly decreases the amount of frataxin protein in cells. A trinucleotide repeat expansion of CTG repeats at the 3' end of the myotonic dystrophy protein kinase gene causes myotonic dystrophy type 1. As with all other trinucleotide repeat expansions, the longer the expansion, the more severe the disorder. The CTG repeat at the 3' end of the gene lengthens the messenger RNA by 5,000 to 6,000 base pairs. This causes problems with the normal processing of the messenger RNA in the nucleus, and it cannot be exported to the cytoplasm. These 4 examples of trinucleotide repeat disorders illustrate that the trinucleotide expansion can occur at any place in a gene and produce an effect. Fragile X syndrome is caused by an expansion in the promoter region of the FMR1 gene, while a trinucleotide repeat in an exon of the Huntington gene causes Huntington's disease. A trinucleotide repeat in an intron of the frataxin gene leads to Friedreich's ataxia, while a trinucleotide repeat at the end of the DMPK1 gene leads to myotonic dystrophy. There are several factors which can confound the interpretation of inheritance patterns. For example, not every disease-causing mutation displays complete penetrance. In disorders with reduced or incomplete penetrance, some persons carrying the mutation do not show signs of the disorder. Reduced penetrance usually is a result of a combination of genetic, environmental, and lifestyle factors which influence the expression of the phenotype. Other disorders can show variable expression, and the range of symptoms can differ in different persons with the same genetic condition. For example, Marfan syndrome, caused by the same mutations in the fibrillin-1 gene, can result in mild symptoms in some persons or much more severe and life-threatening conditions in other persons. In germline mosaicism, a parent's germline is affected by a disease mutation, but their somatic cells are not affected. In this case, normal parents can have 2 or more children with an autosomal dominant disease, even though there is no family history of this disorder. Diseases with the delayed age of onset can produce gaps in family histories, which can confound the deduction of the inheritance patterns in the family. Disorders such as Huntington's disease or polycystic kidney disease do not present until later in life. This means individuals in the family tree that may have carried these mutations may have passed away for reasons unrelated to the disorder, leaving gaps in the family histories. In other conditions, a mutation in the gene can have multiple effects on different aspects of the physiology or anatomy. This can lead to confusing family histories. And the phenomenon of genomic imprinting, in which certain genes are expressed in a parent of origin specific manner. For some genes, only one copy is expressed. Expression of these genes is variable, depending upon which parent the gene came from. In this way, the active gene is preferentially transmitted from one parent over the other. Imprinting does not occur on every chromosome. Only 9 chromosomes are known to have regions of genes that are imprinted. Imprinting occurs by a pattern of methylation, meaning the copy of the gene to be inactivated is coated with methyl groups. This takes place before fertilization in the egg and the sperm cells. The methylation prevents that gene from being expressed. So these genes are often referred to as epigenetically silenced. For some genetic disorders, the expression of the phenotype is dependent on whether the mutation occurs in a gene that is imprinted. In other words, the phenotype depends on whether the mutated allele is inherited from the mother or the father. Prader-Willi and Angelman syndrome are 2 very different disorders, but they both are linked to the same imprinted region on chromosome 15. Some of the genes in this region are silenced in the egg, while others are silenced in the sperm. So someone who inherits a defect on chromosome 15 is missing different active genes depending upon whether the chromosome came from mom or dad. If the defective chromosome 15 is inherited from the father, Prader-Willi syndrome is the result. Symptoms will include learning difficulties, short stature, and a characteristic compulsive eating. If the defective chromosome 15 is inherited from the mother, Angelman syndrome will result. The symptoms include learning difficulties, speech problems, seizures, spastic movements, and an unusually happy disposition, hence the name Angelman.